Cut off your head, you'll know.
Okay.
Okay, so you want to introduce yourself again?
Sure. All right. So good morning, everyone. Thank you again, Ahmed. Thank you, Don, for having me here. My name is Mansoor Amiji. I'm professor and chairman of the Department of Pharmaceutical Sciences at Northeastern University, and I've been on the faculty now for close to 20-some years. It's great to tell you a little bit about some of the work we are doing in the area of nano and especially in focusing on drug delivery, and I'm really glad to see so many friends here today. So first of all, let me just start out with, I know a lot of you in the audience know some of the challenges we deal with when it comes to drug therapy and delivery especially, and I like to put these in three different buckets. The first one really, and I think one of the reasons we are all here today, is to talk about some of the chemical challenges. As you think about the new generation of therapeutics, most of these are either very hydrophobic, meaning some pharmaceutical company and some executives like to call them bricks or sand, because they just do not go into the aqueous media. There is absolutely no way to get these molecules in water. And you can see one of those examples up here. And then on the other end of the spectrum, as we think about more and more of these biomolecules or molecules that are much more specific for diseases that involve nucleic acids or peptides or proteins, these are water soluble. They do dissolve in water, but then they are impermeable. And so in this spectrum of looking at molecules that are very hydrophobic or very hydrophilic, you have basically issues that relate to lack of solubility and being able to be ferried by blood, or on the other end of the spectrum, molecules that are hydrophilic but impermeable and labile. They degrade very quickly, and so they're not reaching their target in the right concentration. The other challenge is, the other challenge is what I call pharmacokinetic challenge or biodistribution challenge, and that's where the targets for our therapies are. Before, as we think about drug delivery in general, we look at blood as our conduit for bringing drugs to the right place in the body. And the targets that we are looking at are becoming more and more remote from the delivery site. So if you think about oral, for instance, if you take a pill, the drug actually goes into the GI tract, it then gets absorbed from the GI tract, and then ultimately through the bloodstream, it travels all over the body. And as it travels all over the body, it needs to actually get to the right target. And as you think about, for example, targets that are inside the cell, like the mitochondria or the endoplasmic reticulum or the nucleus of a cell, that's very remote from the site of delivery. And if you think about how drugs work, these molecules need to reach this target and then be able to then actually stay at that target for a sufficient amount of time to allow you to get the therapeutic effect. And also importantly, that the molecules should not distribute to places where you don't need them to go, what we call off-target effects. And so you're trying to reduce the toxicity of the drug while trying also to improve the therapeutic efficacy. And so this is a huge challenge right now in how do you get specificity of molecules, especially very potent molecules, without having them distribute all over the body and trying to get a lot of non-specific effect. And then the third issue is really how do you make drugs that are currently delivered through maybe a parenteral route, where it is injected into the body and requires a clinical site to administer. How can you use drug delivery to basically try to convert a drug that's invasively administered into the body and make it non-invasive? For example, could you make a system which is administered in a clinical setting, but by using a formulation approach, make it orally bioavailable such that it can actually help patients do self-administer and ultimately improve patient compliance. So we are thinking about all of these three different areas in drug delivery and trying to figure out how do you make sure that molecules that we are discovering are able to actually become drugs. And so how do you address these property issues which are critical in the drug development cycle? So I'll give you three examples of work that, and again, these are just three very illustrative examples in the interest of time. But we tend to work on all of these different areas and think about how we can get therapies to the right place. And the advantage of being in a pharmaceutical sciences department is that we cover the entire spectrum. We go from molecules to patients. We have all the different tools at Northeastern, and you'll hear, my colleagues, for example, in Professor Torchulin's lab and others in the pharmaceutical sciences department, that we can basically formulate, characterize, do in vitro testing, animal models of diseases, do all the pharmacokinetics, all the biodistribution studies, and ultimately come up with the preclinical evaluation of these products. So let me give you an example, again, focusing on these three different areas very briefly. Our first example is how do you improve oral solubility or solubility and oral absorption. I'm delighted Mimi is here because I'm going to plug her technology here. So the idea is you're giving drugs in the gastrointestinal tract, and as I said, about 80 to 85% of these molecules either go into these two ends of the spectrum. One is very hydrophobic, or they tend to be very hydrophilic. And oral route is really one of the most common route of administration of drugs into the body. Every time a new drug is identified, the first question is this orally bioavailable? Because you know that in a pill form and a capsule form, that's going to be most patient compliant. The challenge is that once the drug gets into the GI tract, you have this contraction that is occurring, so there is limited residence time for the drug formulation. Dissolution has to occur in the gastrointestinal fluid, and it's only the soluble drug that actually gets absorbed. And then after it gets absorbed, the first place that it goes to is the liver, and the liver is where the drug gets metabolized. So you have lack of absorption or lack of dissolution, and ultimately then first pass effects due to liver extraction, and all of those issues are limiting how much drug actually gets into the bloodstream. So an interesting observation was that one could then exploit lymphatic systems, and just in terms of the different classes of drugs, the FDA classifies, the USP classifies drugs in these BCS classification system. It's called the biopharmaceutical classification system. And the problem children in these classification systems are really molecules of class 4 that have poor solubility and poor permeability. But if you have drugs that have low solubility, like the class 2 drugs here, but high permeability, at least you can then think about formulation strategy, because you can then improve their absorption if they are able to be permeable. So an interesting approach that we have been exploiting for a number of years now is to look at lipid-based enhancers. How do you then take your drug and combine them with lipid-based nanosystems to try to exploit oral bioavailability? And one approach that is really important is that instead of just looking at the blood as a conduit, let's look at lymphatic system. Can we use oral lymphatics to try to get drugs into the biological system? And one of the advantages of this is really using these lipid-based structures to create an encapsulation nanosystem that then can take advantage of lymphatics and get the drug into the body and bypass the liver. And that's where our nanoemulsion concept comes in. So we have been working, and again, through Mimi and some of our other colleagues, we've been able to actually expand this idea towards manufacturing in a much larger scale. So we use specifically in the IP that we have using omega-3 fatty acid containing edible oil as a way to encapsulate the drug. And so we use natural, edible, very biocompatible materials to try to create these nanoemulsions. And you can see the emulsion droplet size ranges from about 100 nanometer to about 150. These are developed using an ultrasound method. When you go to microfluidic approach and the microfluidizer, which is the technology that Mimi's company makes, we have been able to generate particle size in less than 100 nanometer. And these are reproducibly made. The surface is modified with various types of surface modification, biocompatible surface modifiers. We've used phospholipids. We've used various types of natural polymeric surfactants, as well as synthetic surfactants. And these combination allows us to create a droplet, an oil droplet, basically very similar technology to liposomes, except in the case of liposome, the core is water. In this case, the core is oil. And this oil core allows you to then incorporate inside various hydrophobic drugs. So anything that's insoluble in water gets encapsulated inside. So now you've been able to basically create a vesicle that takes the properties of the oil based on its inherent solubility of these drugs. You can encapsulate inside. Since the drugs are not permeable or do not partition into the aqueous environment, they remain confined within the oil droplet itself. And then the surface of that oil droplet is modified so that you can then allow you to get absorption, targeting, whatever other features that you're looking for, including imaging function as well, if you want to use this for both combination delivery and imaging. And so we have been testing this hypothesis for a number of different applications. Let me give you just one example, or two examples of these. The first one is we take advantage of oral absorption. And one of the challenges in oral absorption is that you have these enzymes that are expressed in the gastrointestinal tract, and as well as transporters that are expressed in the gastrointestinal tract that remove the drug or do not allow the drug to be absorbed. So we took advantage of a very natural, very safe, this is basically Indian spice that is known to affect the expression of these transporters and enzymes. And by using curcumin, we've been able to lower the expression of these efflux transporters. So if you pre-treat the cells or you pre-treat the animals with curcumin, you can decrease the expression of these transporters and enzymes, and you allow more of the drug to be absorbed. In addition, putting the drug in this nanoemulsion gives you better bioavailability because you are able to see much greater amount getting through the lymphatic transport. To test this hypothesis, we use a cancer model. So we use ovarian cancer model to test the idea that could you take a drug that is currently not available orally, and the fact that it's only injectable and it's incredibly insoluble in water, and make it orally bioavailable. So going through that, the three different limitations in drug or challenges in drug delivery. The third one that I talked about, how do you convert a parenteral or injectable product into an oral product? So in this case, we use a drug called paclitaxel, or it's commercially available as Taxol. It's a very hydrophobic drug, log P value of 3.7. Which means that it's literally insoluble in water, and the product that's currently manufactured comes in this combination of chemophore and ethanol. And we took this drug, put it into the emulsion form, and gave it orally. And these are the actual data in animals. If you give in on the left-hand side here, you're seeing the blood levels of this drug given in different formulations, given either as control in just the solution form or as the emulsion form. And what you are able to see is that the area under the curve, which is the indicator of the extent of absorption of these drugs, is about almost in the case with the nanoemulsion, the NE here, and a combination when the curcumin was actually used to pre-treat these animals to lower some of the transporters and these efflux molecules like P-glycoprotein and the enzyme, we're able to get at least threefold higher area under the curve values with the nanoemulsion formulation of paclitaxel given orally compared to the solution form. And because these are tumor models, these animals had ovarian cancer, human ovarian cancer in them. We could actually measure the levels of drug that accumulate in the tumor. So 6 hours after oral administration, we're seeing this black bar here that shows the highest concentration of the drug in the tumor. So taking advantage of this nanoemulsion system, you're able to take a drug that is currently injected into the body and requires clinical facilities to be able to deliver and convert it into an oral product. And we've used this with other types of therapies as well, and shown that it's really the combination of use of these omega-3 containing fatty acid oils, as well as surface that's modified with negatively charged emulsifiers that help you to get lymphatic absorption. And we can also see that this is effective in terms of reducing cancer burden. So the emulsion form in the presence of curcumin is able to give you this tumor volume or decrease in tumor volume compared to all the other controls that we have tested. And we're able to give 2 doses. This is just giving 2 oral doses of the drug at 20 milligram per kilogram, which is the standard dose for paclitaxel. And we're able to see this significant reduction in tumor burden, both in terms of volume and the mass of tumor at the time of sacrifice. So this is one of the approach that one can take advantage of drug delivery system to really try to overcome some of the barriers. Solubility enhancement by using an emulsion and using these lipids in these edible oils to try to dissolve the drug, still maintaining the consistency, it's because it's an emulsion, still has the same aqueous, the external medium is still aqueous, so therefore you are still able to inject this product without any viscosity concerns. The second example, again, using the same idea, is how do you go into some of the most challenging aspects of delivery, and in this case, the brain. We know that diseases of the brain, Alzheimer's, Parkinson's, are some of the biggest challenges that we have. We're interested in analgesic peptides that basically help in treatment of pain. And pain is, especially in the case of chronic pain, we have several different kinds of chronic pain therapies. The opioids are one of the most, the largest right now in terms of use in chronic pain. But the potential of opioids, the small molecule opioids like morphine and hydrocodone and others, is that there's a significant abuse potential. So a lot of companies are interested in developing alternative therapies for pain, especially using the same type of biological molecules. And one of those is this idea of using peptides. The problem is that these peptides do not cross the blood-brain barrier, and therefore they're not effective in getting across. And we are interested in use of these peptides, especially this 4-amino acid peptide called DALDA. Very, very potent. If you can get this to the right place in the brain, it actually has the same potency as morphine. But the problem is that it just doesn't cross the blood-brain barrier. So what we found, interestingly, is that if you take this peptide and put it in this emulsion system and make these emulsions circulate in the blood pool for a long time so that you're not diluted by the, and the peptide doesn't get diluted in the periphery, and the fact that it doesn't get metabolized as quickly, and you're able to have long circulation and long half-life, you can get very high efficient absorption in the brain. And so we took this peptide, it's a very small, and Lipa was a PhD student in my lab, and simply at this nitrogen here, added a fatty acid group. And we went from C2 up to C8 fatty acid, and converted this very hydrophilic molecule into an amphiphilic molecule. And by having an amphiphilic molecule, you got two different properties. One is that it was able to self-assemble, and the self-assembled peptide actually is more stable than individual because it sterically hinders the enzymes and other molecules to degrade it. And then secondly, this lipid-modified peptide was able to go into the oil phase of our emulsion. So we were able to dissolve it into the oil and now protect it from degradation. And so we used this to get, and then looked at levels in the blood. Here you can see the plasma levels. You can notice that on the y-axis, this is a log scale, and this is a standard pharmacokinetic evaluation. It's injected IV, and we're looking to see how much remains in the blood for a long time. And as I said, the real interesting aspect of this formulation is that it has a long circulation half-life. And the fact that you can keep it, this peptide and prevent it from clearing from the blood pool is critical in order to enhance its absorption in the brain. And we showed that, once you get it, you also get into the brain. Because if you can keep the peptide from a long period of time, you can actually get it to permeate across the blood-brain barrier. And the composition of this emulsion, especially the content of the oil, the omega-3 fatty acids, help us to get these molecules buried across. And the size of 100 nanometer or less is critical also in order to get it to pass through the blood-brain barrier. So here is another approach where we are able to take a very simple oil and water and nanoemulsion system, again, manufactured using this microfluidic technology, and you can generate fairly large volumes and even translate this into GMP manufacturing operation that allows us to, get this into commercial scale. And show that we can, by designing it appropriately, engineering all the systems with the right content, right composition, and right surface chemistry, we're able to get this very difficult to deliver drug into one of the most challenging areas of therapy. And then the last example, this was my, some of the other areas that we have actually focused on is in polymeric systems, and we've used those for cancer therapies. We have also been very interested in nucleic acid base, but I'll let in some of our discussions we can talk about our opportunity, but I wanted to end with just some ideas on how, as a group in pharmaceutical sciences, as well as in our own personal lab, how we can collaborate and how we can develop industrial collaborations. And so just some areas that we have done, a lot of these technologies that come out of my lab and other labs in pharmaceutical sciences are protected. As Ahmed said, they're patent protected by Northeastern. And so there's an opportunity for licensing these types of technology right from the academic environment. And we are certainly very interested in a number of different startups have been created from there. We also do a lot of sponsored projects. So if there are areas where technology that you are developing could utilize some of the tools that we have in our labs, we're happy to collaborate with you to develop sponsored project relationships. And we continue to do this with a lot of different companies. And I know, I can give several examples of that. Northeastern also has very, very robust laboratory and instrumentation resources. And I think we are looking for collaboration. We have a company that just recently partnered with us, is interested in vivo imaging. And so we are utilizing some of our expertise in vivo imaging and using animal models of diseases to partner with them so that we can do their studies at Northeastern. We have students. We have graduate students who can work as interns, who can work on different projects. So if you're looking for students who are well trained and able to actually partner with, and so Northeastern is the right place. And then of course those students are also available for full time. And then we're also available for consultancy. Any type that you are interested in, any type of intellectual consultancy, all of us in the faculty are experts in our areas, and we're happy to collaborate with you. So let me end here, and again, I'm happy to answer questions both from those of you who are on the webinar as well as those of you in the room. Thank you.
So any questions from the room while I just change the screen and see if there's any questions in the chat box?
Yeah.
You were talking about using tactile in the motion. Have you used the pegylated forms at all, or added any pegylated get the emulsion?
Yeah. So the question is that we've used Taxol or paclitaxel as a drug in the emulsion form, and do these emulsions actually have any pegylation on them? Yes. So one of the ways that we can surface modify these oil droplets to attach any different kinds of modification. Pegylation is very common. In this case, the way to pegylate these emulsions, DSPE, which is a fatty acid phosphate that anchors onto it. So it's just like liposome, it creates a bilayer. And so you have DSPE PEG. And the PEG 2000 or PEG 2500 is a standard that we have used. And then on the other side, we can also use that PEG as a spacer. And then the tip of that PEG, if it's heterobifunctional PEG, we can actually attach a specific ligand. For instance, if you want to create this emulsion so that it targets a cell, we have some examples where we have used folic acid that's modified through that PEG spacer to actually make it targetable to ovarian cancer cells, or EGFR targeting peptide to target it to a pancreatic cancer cell. So you have the ability to functionalize the surface of these droplets, the oil droplets, based on whatever chemistry. And the good thing about a lot of this is coming from the liposome chemistry. So there's a robust liposome chemistry on the phospholipid side. So we basically take advantage of a lot of things that's already been working in liposome, but instead transfer it into the emulsion.
So have you done oral uptake of those emulsions and the appearance?
Yeah, so we have done oral uptake with Taxol. We have also done oral uptake with sequanavir, which is a protease inhibitor used in HIV therapy. And yeah, in both of cases, it's really molecule agnostic. As long as you can get the molecule to go into the emulsion, the oil phase, your delivery system is what gets you across, so it gets into the lymphatic system. Some of the absorption occurs through the lymphatic system. Some still do occur through the hepatic portal vein, which takes it into the liver. And then after that, it gets into the bloodstream. So we are seeing two different ways of getting it into the bloodstream. And it's really the intact emulsion. We also looked at, after animals have been dosed, we take blood samples at different time points, and we have been able to extract, and it's still the intact emulsion. It's not the drug that's crossing. It's really the emulsion droplet that's crossing and getting us. And we see increase in circulation time. In some of the examples, like sequanavir, we have also seen that if you use these omega-3 fatty acids, we are also able to get better penetration to the brain after oral administration.
So we have a bunch of questions that have come through. I'll just ask you them, and we can answer to the whole group. So is there any nanoproduct for chelation heavy metals in the body for threatening heavy metal toxicosis?
That's a great question. There has been some interest in removing heavy metal. There are two approaches that folks have thought about in terms of removing heavy metal toxicity, using nano formulations, or even one is, you inject these things and somehow use them as scavengers, somehow use them as ways to filter out these toxic materials. And the fact that you get large surface area. The challenge is that in some of these heavy metal toxicity, they tend to be much more, a lot of these heavy metals, especially lead and mercury and others, they tend to be very hydrophobic materials, and therefore there's a very high volume of distribution. They tend to get very rapidly absorbed into the organ. So if you have nanoparticles that can actually get to different organ systems, especially critical organ systems like brain, that would be a much preferred route of getting these toxic material. And then you want to make sure that it chelates and then rapidly is excreted out of the body. So I've seen some work that's been done, especially in case like liposomes have been tried in this area, trying to increase the scavenging potential. But the other approach, which in my opinion, might be a little bit better for removing some of the toxic metals and creating this equilibrium, is in nanofilters and having blood or other biological fluids flow through these filters in order to remove these toxic materials outside of the body, rather than trying to inject them into the body and then expect them to be cleared. Because the challenge with injecting, obviously, you are also exposing the body to the toxicity of whatever material you're injecting, and the alternative approach of actually having them clear by creating a filter may make more sense.
Okay. Another question is, surfactants are known to be toxic to membranes. Tween 80, for example, has been described to disrupt the BBB. Do you have any data concerning the effect of your systems on the integrity of the BBB?
Yes. So the question about Tween 80 as a disruptor of various membranes, it depends on obviously in any of these studies you have to be very careful in interpretation of the results, because what happens is that there are two issues. One is the dose that they administer, and second is the exposure time. And we have obviously done a lot of work to try to understand the potential of any surfactant that's used. So in terms of nanoemulsion, the actual concentration of the surfactant on the surface of the oil droplet is very low. So we're not, and Tween 80 is used in combination with phospholipids. So the total composition of Tween 80 that we use is very, very low compared to some of these studies that have shown that there is actually a disruption in the membrane. In those, those tend to be mostly cell culture studies, and in those systems, you tend to have a very high concentration of these surfactants exposing to the cell membrane, and then subsequently the duration of effect tends to be very high. But in the case of nanoemulsion, we haven't seen any changes in the integrity of the blood-brain barrier with the Tween 80. We actually see the effects mostly because of the presence of the omega-3 fatty acids in the oil.
Okay, great, thanks. How many fold enhancement in solubility of curcumin was observed when converted to Ne?
Oh, that's a great question. So curcumin is incredibly insoluble in water. It's it's again one of those challenging molecules that you cannot get. And in our studies, we find that with doses that we are administering for this PGP downregulation as well as cytochrome C downregulation, we are giving 100 mg per kg of curcumin in these small animals. So these are 20-gram mice. What we have found is that you can get, so when you look at the dose, and you can get fairly high solubility in the emulsions, especially these flaxseed oil or fish oil based emulsions, because they have a very high, in case of fish oil, the omega-3 fatty acid in there is docosahexaenoic acid and docosahexaenoic acid and icosapentaenoic acid. And so these are long-chain fatty acids, and therefore they have a very high value for dissolving hydrophobic molecules. It's called the saponification value of these oils. So that tends to be fairly high, which means that they can actually dissolve nonpolar solutes very well. So yeah, solubility is quite high in these oils.
Okay. The next question is, do I need to contact you or 1 Million Solutions in Health for collaboration? And I'll answer that. I say, why don't you start with 1 Million Solutions in Health, and we'll play the traffic cop and make sure everybody's coordinated from there.
That sounds very good.
Okay. And what about the stability of formed Ne according to ICH conditions?
Right. So we, that's also a very good question. We have tested stability both in terms of stability of the drug that's inside the emulsion, as well as the formulation. So we test the stability based on particle size, coalescence, to make sure that the emulsion remains individual particles and doesn't phase separate into oil and water. And also we have looked at to make sure that the surface composition doesn't change over time. And we have done this study, again, from an academic standpoint, looking at the USP standards and some of the other standards. We have looked at different temperature conditions, and we have looked at different time conditions, and we have done up to 6-month study. I haven't done it, or we haven't really looked at it through the ICH guidelines, but we're happy to collaborate with specific groups that are interested in this and furthering these technologies to be able to do that. One of the applications of these emulsions in the case of ovarian cancer has actually been licensed by a company right here in Worcester, Massachusetts. So they are also testing these with clinical development and working with Fox Chase Cancer Center for that area.
Great, thanks. And last question, then we're going to take a 10-minute break. And I'll ask Mansoor to, while you're having the coffee break, if people ask you other questions, maybe you can just give a quick recap once we start up again in 10 minutes.
Absolutely.
So the last question now is, does nanodrug have side effects in the body? That's the big question.
So, yes. So, I mean, you know, here, obviously, every drug has issues with side effects. And the question that when you think about whether it's nanoformulation or, you know, molecules, whether it's nanoemulsion or polymeric nanoparticles or micelles or liposomes, or different delivery systems that, you know, folks are working on. The key question is, you know, that we try to ask is what's the exposure concentration and what's the duration of exposure? Those are the two major variables that are going to affect, you know, off-target and toxicity of these nanodrugs. In the case of a lot of the drugs that are that we administer, that given IV or orally, the major site is going to be liver. And so most, you know, first question would be, is this drug going to be toxic to the liver? And the you know, when you want to answer that question, the way to think about that is one could be from the molecule side of things. You know, how is this molecule going to behave in the liver? Is it going to actually cause toxicity if it's administered to the liver and there's a high concentration that's reaching the liver? In some cases, the answer may be no. For example, if you are delivering, let's say, a gene construct or a peptide, like in the case of these pain peptides, even if you get drugs in the liver, the fact that the liver is metabolizing these peptides very rapidly, and there's really no receptors for morphine in the liver or for opioids in the liver, you don't see any significant effect of these peptides, even if you're getting high concentration. The second issue is the delivery system itself. You know, when you're getting these, if it's a polymer, for instance, even if it's a biodegradable polymer like PLGA, you may get toxicity from the delivery system itself because you're increasing the concentration and the duration of exposure into a single organ system, which previously, when you're giving free drug, you're not getting as much exposure. So it's really critical to get that assessment early on. And in our case, you know, one of the things that we focused on in a lot of these delivery systems is making sure that we use biocompatible molecules to start with. Even if you engineer them with very sophisticated engineering system, the choice of, you know, edible oils, making sure they're safe, they're metabolizable, polymers that are biodegradable, you know, composition that are, from FDA perspective, are considered generally regarded as safe or GRAS materials. Those are key points to really start to think about. Even though you're testing it in cells and mice, you really need to start with some safe material. And then, you know, you can always add complexity to it as data comes through, but really starting point has to, you know, address some of these topics. Otherwise, it becomes, you cannot translate any of these things further.
Great. Okay, so we're going to take a 10-minute break. For the people in the room, I'm going to put